everybody. Thank you all for coming to the Humanities Center today for the second session in our series on the frozen realms. And if any of you have driven up to campus since 9 o'clock this morning, you'll see that this gorgeous photograph is displayed on the billboard. We are honored to have the photographer himself here, Bryson Patterson, who will be speaking in the series towards the end of the series. Thank you for putting this gorgeous image together. For those of you who were with us last week, you'll recall how Ron Kaufman, who's here, expertly guided us around the world of ice. And he did this from the perspective of an environmental scientist. And we want today to continue that scientific slant. And I always want to pause and say this, that it's a great joy to direct a Humanities Center where there truly is so much presence of scientists in our work and having a conversation between those of us who work in the arts and humanities and those of us who work in the sciences. So this is a project that we're determined to keep continuing and pushing. And we're going to, as I say, we're going to continue that scientific slant today, and we're honored to have with us three distinguished faculty from USD's College of Arts and Sciences who will deepen our understanding of the science of ice and coldness. So in alphabetical order, this is not in speaking order actually, but I can't move away from what I have in my head. Sue Lowery is a professor and chair in the Department of Biology. She received her PhD in marine biology from UCSD's Scripps Institution of Oceanography and has been a recipient of the National Science Foundation Pre-Doctoral Fellowship and a National Research Council Postdoctoral Fellowship. Dr. Lowery's particular research interests lie in the effect of endurance swimming on the development of muscle in juvenile marine fishes. She's been a faculty member at USD since 1990 and is a past recipient of the Davis Award for Teaching Excellence. Michael Mayer joined the USD faculty in 1994 and is professor of biology. He received his PhD in botany from Washington State University, and his research focuses on plant systematics. This focus on plant diversity involves deciphering the evolutionary relationships among plants and then using these patterns to infer the processes by which plants evolve, speciate, and produce new lineages. Dr. Mayer has collaborated with researchers at San Diego State University and the Natural History Museum in Balboa Park. Our third speaker, Maren Mossman, is Claire Booth Luce Assistant Professor of Physics. She received her PhD from Washington State University, and her research involves the creation, manipulation, and study of Bose-Einstein condensates, which is I don't understand any of this, so I'm actually just sort of, but this, I'm told, is the fifth state of matter after solids, liquids, gases, and plasmas. Bose-Einstein condensates, as the coldest form of matter in the universe, give us unprecedented access to the quantum world, and Dr. Mossman uses them to study the fundamental nature of matter, fluid flow, and quantum turbulence. Her interest in ultra-cold atoms is, of course, highly apposite to our theme this semester of the frozen realms, and here the very frozen realms. So please join me in welcoming Sue Lowery, Michael Mayer, and first to speak, Dr. Maren Mossman.
Okay, so today I'm going to be talking to you about the physics of cold, and we're going to take a kind of road trip from classical to quantum, and I'm going to explain to you kind of the types of stuff that I work on, because when I talk about cold, I am in an entirely different regime as when other people talk about cold. All right, so when we talk about something that's cold, we're really talking about, we're thinking about temperature. And temperature is really the amount of kinetic energy something has. So you can imagine temperature as things that move around a lot. There's lots of motion. That's more temperature. And things that move around less, that's less temperature. Okay, but we should really reflect that cold itself is relative. So cold to one scientist might be very different from cold to another scientist. And you know, I always think, you know, when you're sitting at your kitchen table without socks on, and you think that you're doing fine, and then all of a sudden your feet touch something warm, and you're just like, oh wow, my feet were actually very cold. You don't really realize it until you compare it to something else. So it is a relative thing, and that the whole science of cold is really relevant to the discussions that we have in thermodynamics and condensed matter physics and in quantum mechanics. Okay, so for our classical understanding of cold, we have a couple laws of thermodynamics. First law is that energy is conserved. It can't be destroyed. This is a principle in physics that's really great. If something gets colder, that means that the energy had to go somewhere. Okay, it had to either be work put into the system or heat taken out of the system. Okay, our second law of thermodynamics tells us that an isolated system, so a system where nothing is interacting with it at all, is going to always evolve towards a state that has more disorder. Okay, and that this is going to be irreversible in time. So if we think about a cup of ice, okay, and we just leave it out on the counter, we know that that's going to melt in the water, and then over a couple of days, that's going to evaporate into the air, and it's going to be an empty cup eventually. Okay, that process is irreversible unless we do something to it. So we can always take that cup of melted ice and we can put it back into our freezer, and then it can freeze again. But that's something that's putting work into the system. Okay, so it's irreversible in time if we just leave it alone. And so as we drive towards these colder and colder temperatures, the disorder of a state, the amount of, like, how many different availabilities you have and the way that the particles can be made up is going to be reduced. And the thing that I really want to stress is that, you know, I work at temperatures that are very near absolute zero being 0 Kelvin, and that this is the limit of cold. And when I say limit, I don't mean a stopping point. I mean the mathematical definition of a limit, which means that you can approach 0, but you can never quite get there. Okay, so when I talk about limits, I talk about mathematical limits. All right, so here's my relative scale of temperatures. These are the types of temperatures that we have in the universe. Okay, so we have things that go from very, very hot, so about 10 to the 12 Kelvin, down to temperatures that are at the 10 to the minus 12. Okay, so we have the sun's core, Earth's core, pretty hot things. And then we have the condensing point of helium, very near, I think it's 2.7 Kelvin. And then we get colder and colder. But what I want to stress is that our human experience is a tiny sliver of what scientists consider when they talk about temperature. And so our human experience is centered around, you know, around a range of about 100 degrees C. Okay? That's because that's where water exists. And so our lives are unique in that they are centered around this principle that we have to have water to survive. Water in my systems is very bad. So when I do my experiments, I bake out the system so that there is no water, because that's what's going to kill a vacuum system. And I have to do my experiments in high vacuum. Okay, so here's just this relative scale of temperature, just to kind of give you an idea of the types of ranges that scientists talk about when we're just talking about temperature. Okay, so we have our human experience. Why do we want to push farther colder? Like, why do we care about things that are colder than what we can experience here on Earth? Well, when we push things colder, when we change their temperature, then we can change the state of that matter. Okay? And so this can lead to new understandings of material strength. Okay, so some materials, when you make them very cold, they can either become brittle or they can become stronger. And it depends on the way that their composition is made up. And so by making things colder, we can have a better understanding of that. Electrical conductivity generally gets better as we make things colder. Okay? There are certain materials that if we make them very cold, they become superconductors. And it allows current, electrons, to flow without resistance. And room temperature superconductivity is a big push in materials research. When we make things cold, its magnetism also changes. So magnets will become stronger. The properties of the magnet will become stronger as we make them colder. And then chemical reactivity also slows down when we make things colder. And so if we make something colder and we want to watch a reaction happen, well then we would have to make it very cold to be able to do that. In my field, there is an entire branch that's on ultra-cold chemistry, where they're looking at ultra-cold molecules and looking at reactions that are happening and trying to understand the fundamental nature of that. And so by changing the temperature to colder and colder temperatures, we're going to see these changes in states, and we're going to be able to learn more about the fundamental nature of matter, the fundamental nature of the system. And so when we talk about the phases of matter, you probably think of gases, liquids, and solids, and the way that we can go from ice to water to vapor. Okay, so when we think about the states of matter, a lot of times these are the three things that come to mind. If we push to higher energy, if we push to higher temperatures, then we can create something called plasma. That's the fourth state of matter. And shout out to Greg Severn, right here, who knows all about plasmas, okay? But if we want to push colder, we need to start using techniques that are a bit different. So we can push some of these limits by using cryogenics. That's not cold enough for me. So we have to start to push these limits more by using new types of techniques. And so this is where I come to the part of the talk, which is, what is classical mechanics? How do things behave classically? And then what happens as we reduce the temperature? Okay, so if anyone's ever been to one of my physics talks before, you've definitely seen this slide before. I apologize. I love these slides. Okay, so if we think about classical mechanics, if a cop pulls you over in your DeLorean and asks you, how fast were you going? Do you know how fast you were going? You can tell them for sure, yes, I was driving at 87 miles per hour and I am in La Mesa. I know exactly where I am and I know exactly how fast I was going. Okay? And that's because in classical mechanics, we think of matter as being particles, and particles are very well localized. Okay? And we even model, in chemistry, we model particles as being little balls, and we attach them with little spokes to show the chemical bonds. So we think of things as being particles. But then in classical mechanics, we also have this concept of waves. Okay? And waves, we think of these two things as being entirely different. They're fundamentally different, but they're really not. Okay? Waves are a way that we can transfer energy in a system, and there's some really cool properties of waves. So for waves, if we have two drops of water that drop onto a pond, we see ripples spread out. And then if those two intersect, we get what's called interference. And so if we get high troughs and low troughs, that means you are constructively interfering two waves. And if we have nothing there, that means that we are destructively interfering. It means that the trough is intersecting with a peak. Okay? And so we know that from classical mechanics that waves can interfere. And so if I do an experiment, a very classical experiment in physics, I can take two slits that are very narrow, and I can shine light at them. Okay? And you would think that what comes out afterwards is that you're just going to see two strips of light after it. But what we see is we see an interference pattern. And that tells us with certainty that light itself is a wave. Okay? So when we look at this pattern, we see these really bright spots. That's constructive interference. And then the dark spots are destructive interference. Okay, so we know that it's interfering, that it's a wave. But then, that's light. Light's totally different from matter, right? Well, if we repeat that same experiment, and we still do it with light, and we shoot one photon at a time, like just one little bit of light at a time, we still get an interference pattern. And so that kind of you scratch your brain a little bit, and it's like, well, what happens if I do this with electrons? So I'm just going to start shooting electrons through this slit, and I'm going to think that I'm just going to just going to get 2 strips on my screen. Okay, and so this picture, all of those little white dots, those are where electrons have hit the screen. So right now it's just like, not too much going on. But then as I continue to shoot electrons at the screen, I end up getting an interference pattern. Okay, this tells me that the electrons aren't just behaving like particles, but they're also behaving like waves. This concept in physics, this is what we call wave-particle duality, that matter can have the behavior of waves and particles at the same time. And so how do we quantify that? Well, thankfully, back in the 1920s, there was a guy named Lowery, and he quantified this. And this is, okay, an equation. So we have a wavelength, okay? Waves have wavelengths, extent. And then we have something that's we have a constant over momentum. Momentum is proportional to mass and speed, mass and velocity. Okay? So if I have something that's very small, like an electron, well, then it can have a really large wavelength because it's inversely proportional. Okay? But the really cool thing is that I can also take a massive object, like a more massive particle, and I can just make it really cold. And that will do the same thing as what an electron can do. And so it turns out that doing experiments with single electrons is not the easiest thing. Single particles is not great, but I can just take my system and I can cool it down to temperatures that are very, very small, and I can see the same types of physics happen. And so now, this gets us to this idea about particles and waves, this duality allows me to enter into this regime of quantum mechanics. Okay, I try not to use the word realm when I talk about quantum mechanics because movies and I'm not into that. Okay, but we're in this whole different regime that we can study quantum mechanics when we make things really cold. And so now, when my cop car asks how fast we're going to an electron, well, my electron says, well, I don't know how fast I was going, but I know where I am. Okay, and this principle is known as the Heisenberg uncertainty principle. This is Heisenberg down here. And it says that you can know your position and your momentum or your speed to a very certain amount of precision. Okay, you can never know one exactly. All right, so if we push farther into the cold, then we reach the next phase of matter. And this is a dilute gas Bose-Einstein condensate. So this over here is a picture of going from a thermal cloud all the way to a Bose-Einstein condensate. And those were taken on the International Space Station, which is pretty cool. Okay. So one thing that's really interesting about Bose-Einstein condensates is that if you think about it from a thermodynamic perspective, it should be forbidden to get to that state of matter. It should be forbidden. But just because something's forbidden doesn't mean it's impossible. And the fact that it's forbidden makes it fun to study, because it is possible as long as you are very precise and very careful about cooling down your system. So these dilute gas Bose-Einstein condensates, this is what we call ultra-cold physics. And so we start with high temperatures, and the matter is going to behave classically like particles. They're billiard balls bouncing off of each other. And then we start to cool down the system, and as they cool, their wave nature becomes larger. And then as you cool farther, they become larger and larger, and eventually you end up having a very large wavelength, and they all sit on top of each other, and it becomes something that is now macroscopic. Okay? When I say macroscopic, that means that I can take a picture of it. That, like those pictures right here, these are taken just with a regular camera. This isn't a microscope that they're using to take those pictures. Okay, you don't because this is millions of atoms that are just that have the same wavelength, and they're behaving like what some people would call like a super atom. All right, so we get down to temperatures that are very, very cold, very near absolute zero. I'm talking about 50 nanokelvin. Okay, that's 50 times 10 to the minus 9 Kelvin. That's pretty cold. I don't know, I'd say that's pretty cold. But it's really hard for us to imagine what that actually means. Like, orders of magnitude are really hard for us to understand. And so, to better understand this, I use my length comparison. So if I go from LA to New York City, that's approximately 4,000 kilometers. Okay? I'm going to say that that's equivalent from absolute zero to room temperature. Okay? So I make that comparison, and I see that for something that's 50 nanokelvins, it's about 0.7 millimeters. So that would be like standing in LA and being a grain of sand away from your absolute zero. So we're talking about something that's extremely cold compared to room temperature. Okay. Achieving these temperatures can be very challenging. It's not easy to do. Like I said, it should be forbidden. Okay? But again, that's what makes it fun. Okay, so the limitations of this type of research is that you need an ultra-high vacuum. You can't do this just in ambient air. You need lots of lasers. You need lots of equipment. Like this is my apparatus. This is me opening up my ion pump, and then I have to bake it out so that I get all the water out of the system, because water's a killer for vacuum systems. I need to have extreme control over everything in my system, because the more energy I take out from my system, the harder it gets. Remember, absolute zero is a limit. You can never quite get there, but to get there, you need to keep pulling energy out of the system. And it takes a lot of time to build. So it takes a lot of man hours and a lot of time and dedication and blood, sweat, and tears. Okay? There is a current push to make these systems commercially available. One of the grants that I just got is to use some of the systems that are in Boulder, Colorado, to be able to do some of this research remotely, which is pretty awesome. And we also do this on the International Space Station, which is also pretty awesome that we do that. Okay? So it's really hard to do, but the things we can learn from ultra-cold physics are so many. So I work in a field that's known as quantum simulation. So what I do is I use a quantum system to simulate other systems and learn about the fundamental nature of how those systems work. And so there are a lot of different things that we can learn. I focus primarily on superfluid dynamics, materials design, the fundamentals of quantum mechanics. There are a lot of different things that you can do. There are other types of cold physics experiments. You know, we have ion trapping, cold chemistry is one that I mentioned earlier, and then metrology, which is like atomic clocks and things like that. And you know, this is just a time sequence of an experiment that I have done. You know, this is a picture of a Bose-Einstein condensate in action. All right. So the takeaways that I would like you to have from my talk are that cold is relative. It really depends on who you're talking to. Our human experience is very unique because it revolves around the fact that we need water to be able to survive. And even though we live in the classical world, underlying all of this are the laws of quantum mechanics. And the quantum world really does have the key to understanding the fundamentals of nature and the fundamentals of just life itself. So if you would like to chat more about quantum mechanics, you can always email me. But we also have a physics coffee hour every Monday from 12:30 to 1:30 in the third floor atrium of Shiley. And I make fresh, homemade baked goods every week. And if you can either bring a mug, because we make a big vat of coffee, or you can
So I'm going to get a little warmer than that, actually quite a bit warmer than that. And so we're going to talk about animal adaptations to ice and coldness. So I want you to, since I unfortunately already started with a picture, close your eyes and think about either the Antarctic or the Arctic, and think about what kind of animals you would see. Now open your eyes, and how many of you thought about a mammal? Arctic fox, hare, something like that. Polar bear. How many of you thought about a bird? Like my penguin, since I see you in this room. So probably you were thinking about, a homeotherm. And a homeotherm is like birds and mammals, organisms that can maintain a constant body temperature regardless of what the temperature is outside, within limits, not as cold as minus nanokelvin. So how do they do that? So if we look at our seal that's in the breathing hole there, insulation. So seals, whales have a lot of fat, and they use that blubber in order to prevent the heat loss that they've made within their bodies. The macaques on the bottom there are exhibiting another great mammal characteristic. They're covered with fur, and when they get cold, little muscles at the base of each one of the hairs makes them stand up, and you get a great insulating layer of air underneath the fur, and you stay warm. We have remnants of this. How many of you have had goosebumps? That is our attempt to warm up by lifting our exceedingly limited pelage. And then you've got penguins and the other birds, which have feathers, which are also great insulators and can keep a lot of that heat in. You can also increase your metabolic heat production. Shivering, we've probably all experienced that. Having those muscle contractions that don't contribute to locomotion means that energy is going to be dissipated as heat, and then you can capture that and use that to warm up. And then I'm going to talk in a minute about some non-shivering thermogenesis. You can use regional heterothermy. So in other words, you can have parts of the body that are warmer than others, and then you can control where the heat is kept so that you can keep important things warm. And then, of course, you can use behavior. So the penguins on the lower left there are huddling together, and this helps them to prevent some of the heat loss because they're one large mass instead of individual. And there's other kinds of behavior that your favorite animal might be using. So let's talk a little bit about some of the ones that you might not be so familiar with. For instance, non-shivering thermogenesis. And the structure that accomplishes this is brown adipose tissue or brown fat. Now, this is not the fat that we're used to being concerned about. So it's not your beer belly. It's not your love handles. It's none of that you might go to liposuction for. These are very tiny little islands of fat cells that have very different structure and very different metabolism. And so what they can do is they have an uncoupling protein which inserts into the membrane of the mitochondria and disrupts the normal flow of electrons that produce ATP, chemical energy currency. And without building up that ATP, you lose the feedback, so you keep burning energy and you keep creating heat. And so originally they were found in newborns and in hibernating animals, and they use it to come out of hibernation to generate the heat for that. But in the early 2000s, they discovered that adult humans have these too. And so they're these tiny little islets that you can see in the dark structures on the left. Well, how do you actually show that they're not just weird fat, but that they actually do something like generating heat? Well, you can use something like a PET scan, a positron emission tomography, where you use an isotope of glucose. And so it gets concentrated in areas that have very, very high metabolism. So if you look at this image on the left, that's a lean person, so not too fat, and they're in a thermoneutral zone. So in other words, they're at a temperature that's comfortable. They don't have to expend any extra energy to maintain that body core temperature. And you can see that there are not very many dark spots. However, if you take that person, put them in a cold room, or like a meat locker, and without a jacket, and you leave them there for a while, and then you bring them back and put them back into the PET scan, then you would see that all of that, you would get very high activity overlapping with all of those zones of the brown fat. So showing that it becomes very active when you need a little boost of temperature. Now the regional heterothermy is what allows these penguins to stand on an ice floe and not get frostbite, or lose their body temperature. So if you look at them, they have a structure called a reap mirabile, which is a wondrous net of blood vessels, thousands and thousands of loops that are in the appendage. And so they would be, say, in the flipper or in the foot. And these side-by-side loops of blood vessels allow the recovery of heat as the blood is circulating from the body down into the extremities. So when the blood is leaving the penguin's body and it reaches the feet, and you can see that you lose the feathers for insulation, so you're going to be losing heat as that blood flows into the feet, which are on that very conducting ice flow or cold rock. And what we see is that the arterial blood is always warmer than the blood that's returning in the adjacent location, and so you get a diffusion of the heat. So the temperature is already cold when you get to the extremity, so you're not losing that much heat by conduction. And then as it comes back up, it picks up the heat as it goes back into the body, so you've conserved heat so that you reduce your energy costs of standing around in the ice, like with an egg on your foot. And it also means that you don't get frostbite, because you're still circulating the blood to the extremities. Now, humans lack this wonderful net in their extremities. And so if I were to stand on an iceberg barefoot, then I would also have regional heterothermy. I would just reduce the blood flow to my extremities, preserving the heat for my heart, my brain, my lungs, all those important things, and just ignoring those feet, which I might not need to stay alive. However, without the blood, we're going to have no oxygen delivered, and you get all kinds of havoc with cells. And so they start to die, you get frostbite, then you get cold, and you freeze. And then you can have tissue death and everything that follows. So it's a very big challenge for humans to be in the polar regions. And so I'm not going to talk about how humans have adapted to the polar regions, because we really aren't as interesting as these other animals. And this is an expedition that's the last one that I know of that went to the North Pole. And it was a really interesting one. It was an all-female group with, I think, more than half of the women were from Arab countries. And so they were the first Arabs to go to the North Pole, first Muslims. And maybe it was the first all-women team, too. But if you look at their gear, they have to make up for the fact that we are not evolved for living in the poles. So whereas Perry, in 1909, the first Westerners to reach the North Pole, they would have been covered with furs from other animals in order to reduce that heat loss. Now we have all kinds of fancy materials, but they still face those difficulties of frostbite and other things. So what about the animals that can't maintain a body temperature and just avoid freezing because they're not cold? So like our Antarctic ice fish here, it's in seawater that's actually negative 1.9. So how does it avoid freezing? So that's what we're going to talk about. Well, ice is the part that really is damaging. So normally you don't find ice forming low in the seawater, but if you want to watch that happen, look at this BBC video of a brine sickle forming. And so what happens is you can actually see this icicle that's very, very dense, salty seawater forming, and it goes down from the surface sea ice to the shallow floor there, and then goes across the ocean floor and freezes everything in sight. And so instantly, all of these sea stars would be covered with ice and then probably freeze. Well, what's so bad about freezing? I mean, we freeze things all the time to preserve them, right? Well, if you want them to be alive at the end, it's a much trickier process. So if we look at our frog here that's got the ice crystals forming on it, we can sort of think about what might be happening to its cells. Membrane and proteins are compromised, their flexibility is reduced, and they don't function very well. The extracellular ice formation, and so that would be the ice outside of the cells in between, because we still have fluid components between our cells. And so if you get ice forming there, as the ice forms the crystals, it recruits water molecules from the moving part of the fluid, and they become part of that static ice crystal. And so then the number of water molecules left behind is lower. So the cell then loses water to the outside as it rushes out, and the ice then gets recruited onto an ice crystal. And so you get a dehydration in the cell, and then you can get damage to the membrane and cell death. If you get ice crystals forming on the inside, they're very sharp, they can disrupt the membrane, and you get cell death. So ice crystals, not so good, no matter how beautiful they are. These frogs can actually freeze and then be reanimated. So what happens to your membranes? So now I want you to think about a stick of butter. Okay, I know it's not a great analogy, but it's certainly something you have experience with. If you, like me, I bake a lot, so I buy bulk butter. So I have a freezer full of butter. And when I pull the stick out, it's very rigid. Okay, so think about that. Even if you pull it out of the refrigerator, it's very rigid. All of those lipid molecules have been pressed together, and their molecular motion is quite low. So they're rigid and they're not very fluid. Think about then, if you leave the butter on your kitchen counter, and now, since it's probably about 68 or 70 degrees Fahrenheit in your kitchen, it would be softer, but it would still look like a stick of butter. So it would probably be in that kind of middle range where you've got enough movement that if you had proteins suspended in it, and they were enzymes or something, they could move around, they could function, and do their thing, but you'd still have integrity. Now in the summer, my kitchen gets to be about 85, and my stick of butter melts. And so then it gets too fluid. All of those phospholipids are swirling around and liquid, and then that's not very good for your integrity of membrane either. That's for the desert talk. But this is for the cold side, we'll be thinking about the rigidity. So what's a poor ice fish to do? Well, you can substitute lipids so that you maintain a soft and flexible membrane. So you can throw in polyunsaturated lipids into your mix, and just as if you were to mix olive oil or vegetable oil with your butter and then put it in the refrigerator, it would remain nice and soft and malleable, and your membranes can continue to transmit nerve impulses, do ion transport, all kinds of things it needs to do. There's a limit to how much you can improve the function of a membrane with cold temperatures. So at some point, it is going to be too rigid, and also if there's ice crystals, you get expansion of the size and the size of the cell, and then that can rupture the membrane. So how can you avoid freezing? So typically what organisms do is they produce some sort of a cryoprotectant. So a protectant is going to help them offset whatever negative effects they have, and cryo is the word that's associated with freezing or with cold temperatures. They can make small molecules that are just additive, or they can make some special types of molecules that actually decorate the surfaces of the ice crystals and prevent them from growing. They can, and these usually have a property where the melting temperature and the freezing temperature are not the same, which can be useful. You can have supercooling of the fluids, in which you have little molecules that can nucleate ice crystals where you want them and not where you don't want them, and then also control their size. Or you can actually have some sort of freezing tolerance. And for many organisms, this is a seasonal thing. So if you're an Arctic flounder, and so it's in the Arctic, so in the winter, you'd be going into November, December, and March. They upregulate the amount of this antifreeze protein in the plasma, and you can see at the same time it allows them to go to much, much colder temperatures before they would freeze. Hopefully, the freezing temperature of the plasma is lower than the actual temperature of the water that they're in. So how these work in a stylized way is, imagine that this is my cell, and all the little blue dots are water molecules. These larger ones and the green ones are other molecules that are in there. This is a growing ice crystal on the outside of the cell. So as this gets larger, it's going to be pulling the water out of the cell. And then over here, we see that overall, there's not as much free water to participate in metabolism because it's all bound up in these large ice crystals. And so we have dehydrated cells, and you can see that they've both shrunk and that the membrane is no longer functional and smooth. Well, you can offset part of that if you make a bunch of these purple molecules, which are the cryoprotectants. And they get in the way of the hydrogen bonding of the water so that you don't produce the ice crystals, and so they don't grow. And then if you have other things that are ice blockers, and these are the antifreeze proteins, they can be on the surface of the ice and then prevent it from growing. And here's one of our wood frogs, and it uses glucose to do this. So if we were to look at a control temperature, let's say September, it would have very little glucose in its legs, hearts, or liver. But if you cooled the frog down in the laboratory, we would see that it would start to manufacture extra glucose and sequester it in the tissues. But if you captured them from the field, you get an even stronger stimulus of this response, and you can see that they accumulate ridiculous amounts of glucose. Now, aquatic organisms can't do this because they lose the glucose into the water, but terrestrial organisms can. So aquatic organisms often are using these antifreeze proteins, and here in our ice fish in Antarctica, if we were to look at its plasma, we'd see a whole bunch of tiny little ice crystals that were decorated with these antifreeze proteins, either on the prism sides or on the basal sides, and it prevents them from getting very big. So they keep them small, they don't grow into these really large snowflake sort of things. And antifreeze proteins are really, really good. And so what we see is that they have evolved multiple times. So on the left, we have a couple of antifreeze proteins from fish, and on the right three are from insects. And evolution works this way. You have a protein or a gene that gets duplicated from some mistake in the cell. And so now you have two copies. One copy that keeps doing whatever it was doing, another copy that can accumulate mutations. And if some of those mutations make them bind to ice, really cool. And so you can select, they can be selected over time, and we see that there's convergent evolution from a whole bunch of different starting points, so that these antifreeze proteins are characterized by a flat surface that is loaded with threonine, which is here shown as the pale blue. That can interface with an ice molecule, and that's how it binds and prevents it from growing. And then down here you can see, if you've got the antifreeze protein, you've got tiny little ice crystals that don't grow. In the other cells, you could have bigger ones, and then these small ones can coalesce and join each other and form really big ones. Well, this is great for the non-human animals because many of them can survive, and this actually means that insects can be found in the polar region, well, mostly in the Arctic, but even in pretty extreme areas. But could we take what their techniques are and take advantage of them to solve some problems that humans have? Organ transplant is a very difficult thing to manage, because once we remove an organ from a donor, these complex tissues don't last very long. They have a very short lifetime, and even if you put them on ice, they don't last very long, and you can't freeze them, at least with the techniques that we have now. So you can't save them until you find a match, and the people have to live close together. So people have been experimenting with using some of these cryoprotectants that have been developed naturally, and if you combine them with some cryoprotectants and some techniques where you cool at a certain rate and then thaw at a certain rate, we're working towards being able to freeze long-term these organs. Now, we can freeze small things. There's all kinds of sperm banks. There's oocytes that can be, you can even freeze embryos. But things that are complex tissues with neurons and with blood vessels, we're not so good at that anymore. Now, on the top, this is a rabbit kidney, and so the sutures, or the black things, it's not a spider. And on the left, you see a kidney that's been frozen, like with just some regular lab solutions, and it's a solid block of ice. You can't freeze that and transplant it into another organism. On the right, you have a kidney that's been treated with some of these cryoprotectants and antifreezes and undergoes a special cooling technique so that its cells have actually vitrified. So instead of freezing into ice crystals, they are now sort of immobilized. The molecules are immobilized, so they're a glass-like lattice, and translational molecular motion is ceased, or very close to ceased. And so what that means is you don't get any volume changes, you don't get any osmotic changes, and so if you thaw it out properly, then you should be able to transplant that kidney into another animal and see that it works. Still under development. And I just wanted to end by saying there's a lot of variation in human susceptibility to cold. I wanted to point out our friend Ron Kaufman in the Antarctic with his shirt sleeves. Normal other members of the crew with a jacket on, and then I have two.
thanks once again for the invitation to participate in this great program. And what a fun group. I'm going to borrow heavily on Sue's talk, which is really convenient for me. I'm going to talk about plants in the frozen realm. And let's see here. I'm going to use just the space bar, I think. What I'd like to do today is talk about mostly the poles, mostly the Antarctic and the Arctic polar regions. It will simplify the discussion a little bit. And of course, how can plants live there, and how are the polar regions different from each other, and how and why are their floras different? And here's a gratuitous picture of the Northern Lights. Have nothing to do with my talk. So we think about what plants need and where they can be. Obviously, we know things like they need sunlight because they photosynthesize, and they need air that contains oxygen and carbon dioxide for metabolism and for photosynthesis. They need some nutrients to build molecules, but also they need water, liquid water, right? And that's an issue for the frozen realm, and especially for the polar regions. And I have, just to make it a little classy for this humanities crowd, I've included a poem here, Water, Water Everywhere, and all the boards did shrink, water, water everywhere, nor any drop to drink. Applies to sailors crossing the ocean and plants that are trying to live in a place without available water. So what's the issue with cold? As we already know, it's a challenge. It slows and stops molecular processes, chemical reactions like photosynthesis and respiration. Frozen water is not usable, and it's even dangerous. As ice crystals form, it breaks, disrupts membranes, and pierces tissues and such. So it's a problem for plants, anything that is exposed to low temperatures. But let's look on the bright side of snow and ice. It can insulate organisms, particularly plants. It allows light penetration, so in certain circumstances, plants can photosynthesize below ice and snow and continue on. And it provides very important cues for the life cycle of certain plants, whether treatment of seeds or triggers through that cold process the timing for growth and flowering. And we know this. I mean, temperate plants have adapted to periodic cold, periodic severe cold. We use this constantly in agriculture. This is an example of winter wheat. And this is planted in the fall. It starts to grow, and then it goes into dormancy over the winter. And farmers like nothing better than to cover it with some snow so that it's protected from even the more extreme temperatures of the air and the forces of the wind. Plant adaptations to cold, windy, and dry conditions. And let's remember that there is a great deal of similarity between the frozen realm and the desert, right? Because of the lack of available water. It's a very dry kind of environment. Stimulates plants to grow in a low, dense clump or type of mat sort of form. Surfaces become hairy. That is not only good for insulating and breaking up the wind that approaches the epidermis, but it also reflects the intense sunlight that is imposed on these plants during the summer growing months. We also have protective pigments. Again, so much intensity of solar radiation is a problem, and so extra pigmentation is necessary in leaves. We have the antifreeze molecules. These are not exactly the proteins Sue is talking about, but these are the cryoprotectant acting as a way to allow chemical processes to occur below the freezing temperature. Let's see, specialized photosynthetic and metabolic process. So yes, there's been adaptation of these plants to grow in these climates. And also they can go into dormancy and hide under the snow or hide under the level of, or the soil level in the winter. So with that general background, I want to think about the poles and how they're different. And clearly, right off the bat, we know that the South Pole is occupied by a continent, whereas the North Pole is ocean, open ocean, unless there's ice there. So within the Antarctic Circle is primarily the source of land, except this land is covered by an ice sheet, with the exception of the edges, the very edges of the continent near the water, where there is some available land periodically for plant growth. Surrounding the Arctic Circle is open ocean. Open ocean that allows then a circumpolar current. It just goes round and around and around. Current getting colder and colder, keeping warm water out and keeping the continent even colder. Now, the Arctic region is balmy by comparison, at least seasonally. The Arctic Circle, as you can see, runs through a fair amount of landmass. Going beyond the circle, you get even more land. There's a lot of land that is straddled by the Arctic Circle. And this land is seasonally available, harboring low elevation tundra that allows for a growing season every summer. Low, spongy, swampy, marshy type of permafrost that melts just the top layer, leaving permafrost still frozen below it, but allowing then life to proliferate in this area, both plant and animal. So a vast amount of land available for growth of plants in the growing season of the summer, whereas in the Antarctic, the best it gets is this area along the rim of the continent and that isolation from the rest of the world. So it's perhaps not surprising there are more plants that grow in the Arctic region. There are perhaps about 2,200 vascular plants. And when we say vascular plants, we're talking about ferns and angios, or flowering plants, and conifers in this region. I want to go back. So a lot of available land, like I said, a lot less land, a lot less hospitable climate in the Antarctic, and whereas we have 2,200 vascular plants growing in the Arctic, we have less in the Arctic. What's that number? Ron, you remember that number? Two vascular plants that are native to the Antarctic, and there it is. This was the punchline that I gave away accidentally. You didn't even notice. But there it is. They're both there. The whole flora is there for you. The vascular plant flora consists of Antarctic pearlwort, little cushion-like plant. It is a relative of the carnations, sweet williams, those kinds of the guys, silene, if you're familiar with that. And we also have Antarctic hairgrass, Deschamps antarctica, growing also as a fairly low cushion. There is not really any fantastic story surrounding these guys. They also grow in other parts of the world. The Colebanthus grows in the Andes. Somehow it is able to move, or perhaps it's a relic and it's always been around there, but I'm not sure about that. And Deschampsia is part of a larger group that is found throughout the world. But in terms of adaptation, they use most of the adaptations I've already talked about with regard to adaptation to the cold. But, you know, when we talk about the flora of the polar region, and I mentioned vascular plants, I'm actually leaving out a whole other group of plants that grow there. And these include the bryophytes. And bryophytes are non-vascular plants. They include things like mosses and liverworts and hornworts. I'm really going to be talking about the mosses for a few minutes. The mosses. These lower, quote unquote, lower plants, humble, and we know about them. They grow on the north sides of our buildings in places where it's moist for the most part. And they're an early group to have evolved on the earth and then were later dominated by vascular plants. We kind of think about the characteristics of the mosses as being liabilities when it comes to competing with the bigger plants. What do I mean by this? They think it's, we usually simply assume that their lack of vascular tissue leaves them at a disadvantage. They get crowded out, they get shaded out, muscled out of habitats, and are limited to places here and there. This leaves them, in general, small, because they don't have vascular tissue. They're small, not stiff and woody. So they're very small, they can't get very large. They need a close contact with water. They don't have vascular tissue. They don't have tubes moving water and nutrients throughout the plant body. It actually has to be absorbed through the plant body itself, the surface. So they need always to have some water around if they're going to be active. They don't have true roots. Man, they don't need true roots because they're not pulling up water. They don't have vascular tissue, so they don't have true roots. Another thing is simply they have very thin leaves. And so our thought about moss is that they are these oddities, these delicate little plants that have to be in a very humid environment. Except that it's not really the case. But one more little explanation here. The thin leaves of these mosses they are thin. They are one cell layer thick in many cases. Whereas plants like this is not a heavily adapted desert plant. This is a relative of the lily that actually grows in some of our pots around here. It's the hyacinth. It has many layers, but it has a lot of volume that can hold water. It protects that water by having a dense cuticle, which seals in the plant, protecting it sometimes from sunlight with pigment, but simply from temperature extremes and from evaporation. So all ready to hang on to its water. What is this moss going to do? Particularly if it's growing in a cold region. Well, let's turn some of these characteristics around. What may have been a liability actually might be an advantage in some kinds of habitats. Being a small clumper type of organism is actually an advantage in terms of growth form, right? Close contact with water. Well, when you're sitting under ice and snow annually, you have melting coming up right down and bathing you in water for as long as that water is available. The roots are not necessary because you're not pulling up water. The water is above you. The available water is coming from above you. Thin leaves, perfect for the absorption of water when it's available. But that still doesn't answer the question of why single-celled leaves with no cuticle is a good idea. Well, because mosses have what I'm calling a superpower, because they can become dehydrated and they can go dormant when water is not available. And sometimes it thinks maybe indefinitely, maybe not just overnight or over a season, but maybe many years. Mosses, wherever they grow, including the desert, have mastered sort of this skill of drying without dying. Their thin leaves are not a liability because they tend to simply track the ambient humidity of the air and of the environment. So as the environment is moist, it pulls that water in and keeps everything going. As the environment starts to dry out, so does the leaf. But as simultaneously with the drying process is the building up of these cryoprotectants. And in this case, it's increasing concentrations of sugar-related molecules like fructans and sugar alcohol, mannitol, sorbitol. Molecules that prevent ice from forming and protecting the remaining structures in the cells as they go into essentially suspended animation for months or years. So a little side note here, let's think about the mosses. They were the first first plant on land, 470 million years ago. They made it out onto land first. What's even more interesting, I find, is that the world was simply mossy for 50 million years. They were all alone, right? Just a mossy world. That's it. They lived through lots of ice ages, lots and lots of ice ages, lots of kinds of environmental conditions. Their spores can blow all over the world, and they developed some pretty cool adaptations to be able to deal with the periodic, or perhaps the general lack of moisture in certain areas. But it's not as if they grow luxuriantly. Well, they kind of do, but it takes a while, right? Here are some clumps of moss. Here's this portion pulled out over here. Here's shown here. Because they don't have vascular tissue, the photosynthesis that takes place out on the tips, it pretty much stays there. It isn't conducted through the rest of the plant body. This is convenient for carbon-14 dating because that means all the carbon that is taken in by photosynthesis stays there. So we can age the plant and find out how old this clump is. In some cases, they can be 500 years old, growing 1 or 2 millimeters per year. There are, as I think I said, about 100 species of mosses on Antarctica, about 30 liverworts. These clumps also form a sort of a basis for a whole ecosystem. They are been called the old growth forests of the Antarctic. These dense clumps, if you start working your way through it, you will find bacteria, you'll find fungi, you'll find algae, and you'll find invertebrates of various kinds, including the indomitable tardigrade, the water bear. Truly scary. And then this is, if we're thinking about Ant-Man, here's where the quantum realm
Oh, gosh.
Remember Ant-Man having to watch out for the tardigrades. That was great. All right. Coring some of these regions in these big moss banks have revealed moss that can grow from a block of ice that's been dated at 1,530 years old. So coming back from all that time, suspended animation, freeze-dried, whatever you want to call it, it's ready to go again. All right. Oh, and this is just a cool little story. Glacier mice. So, what you see here, not the trouble with tribbles, trifle from Star Trek, these are independent little balls of moss. They can include several different species of moss. They can include all those other characters that I just mentioned living in this little ecosystem. The temperature inside these balls can be 20 degrees greater than the external temperature. But even better, they move. And they move as a group. Herds, and that's why they call it herds of glacial mice. They were described in the 1950s by an Icelander. And they've watched these things. They've actually tracked them. And they tend to move together, not related to just slipping down ice and not related to the wind. What they do know is that as the glacial ice around it melts, they're basically insulating the ice below it, which leaves them on a little pedestal, which then allows them at some point to roll off. So they roll constantly enough to where all of the ball gets some sunlight. They don't need to be rooted into any substrate. I mean, that is amazing. They roll around and they move together. It's still mysterious, believe it or not. They haven't figured out exactly what is going on, but they get started on a little pebble, and it just grows from there, and they start bouncing around on the surface of a glacier. All right. I think I'm about done. I was going to leave you with a nice picture of mosses, but this penguin walked right through the shot. So that's all I have. Thank you very much.